Assalamu alaikum. Dear students, I'm Farheen Yamin, working as co-teacher in Department of Botany, University of Education. Today, we are going to discuss a very important topic from plant systematics, anatomy, and development. The course code for this subject is BOTN 1112. The topic for the discussion today is structure and development of ovule and its types. As the topic indicates that it is related to ovule, that means here we will discuss about the female reproductive portions of the flower of the plant. How the female portions are developed and what are the types, and here are the contents for the topic. First of all, in this presentation we will be familiarize ourselves with the concept of the ovule. Next we will see the structure of the ovule, representing that what are the parts of the ovule. Then we will see the types of the ovules, and finally we will discuss the development of the ovule. First of all, the ovule. More technically, if we define ovule, then it is an integumented, indehiscent megasporangium. Ovules is a structure that ultimately leads to the formation of seeds. We know that a fertilized ovule is known as a seed. That means ovules can be considered as precursors to seeds. These are also considered as immature seeds. Ovules contain sporogenous tissue in which megaspores develop, which ultimately form megasporophyte. Ovule is very important for the plant propagation and food production. Now, if we are talking about the ovule, we have said that it is integumented, indehiscent megasporangium. Now the question arises that what is megasporangium? Megasporangium is a structure that contains megaspores or female spores in it. If we see the structure of the ovule, then several parts are observed. Ovule is a structure which is actually born on ovary on a specialized tissue which is called as placenta. This placenta is supplied with the vascular tissue. Now we will see the parts of the ovule one by one. The most important or major parts of the ovule includes a specialized tissue, a central tissue, which is known as nucellus, which is also called as megasporangium. This is the tissue within which the female gametophyte develops. This nucellus tissue is surrounded by integuments. Generally, these integuments are in the form of two layers, or we can say there are generally two integuments around the nucellus, and out of these two integuments, the inner integument forms first, then the outer integument. However, there are some other plants in which the number of integuments vary, and where there is one or single integument. The third important portion of the ovule is the funiculus or funicle. Funiculus is the stalk of the ovule. The fourth one important part is the micropile. Micropile is in fact an opening in the integument through which pollen or a pollen tube enters. Then we have a raphe. This structure is actually a ridge on the seed coat, often formed from an adnate funicle. Then we have hilum. Hilum is the place of attachment of the funicle with the central body or main body of the ovule. And finally we have chalaza. Chalaza refers to a point of junction of integuments with the nucellus. Here I have a diagram that is showing all parts of the ovule. Here you can observe the main portion, the main central region or central tissue, which is nucellus, and it is surrounded by two integuments, and along with next you can see the third portion, that was funiculus, that is the stalk of the ovule. Then we have here micropile. As we said that this is the opening in the integument through which pollen tube enters. And then we have a portion hilum. This hilum is the place of attachment of the funicle with the main body of the ovule. And then we have chalaza, which is the junction of integuments and the nucellus, and here we have that ridge which is known as raphe. So these are the basic components or portions or parts of a typical ovule. Next, if we see the structure of the ovule, we have a large cell in the mature ovule embedded in the nucellus, which is known as embryo sac. Embryo sac is actually an enlarged megaspore that contains three small cells towards callosal end, which are known as antipodal cells. It contains a set of three cells at micropilar end, which is known as egg apparatus. And out of this egg apparatus, two cells are known as synergids and one cell is known as egg. And in center of the embryo sac, secondary nucleus is present. Here is a figure in which we can observe all these parts of the embryo sac. Here you can see the three antipodals, and these are 3 cells of the egg apparatus. Out of these two are synergids and this one is egg, and in the center of the embryo sac there are present polar nuclei. So this was all about the parts of the ovule. Now we are going to discuss types of ovule. Ovule type is primarily based on the curvature or bending of the funicle and nucellus. It is based on the degree of the curvature of the funicle. On this basis, we have 4 to 5 different types of ovules. The first type is orthotropous or straight ovule. As the name indicates, in this case the ovule is straight. Here you can see the ovule is straight and upright on placental surface. In this case, micropile, chalaza and funicle, these are in a straight line. Such type of ovule is known as orthotropous or straight ovule. Such type of ovules are found in the members of Polygonaceae. The second type of ovule is anatropous. In anatropous ovule, the ovule is inverted, as you can observe here. The whole portion is inverted here, bent, curved here, and it is adnate to funicle. In this case, the micropile faces the placenta. Such type of ovule, inverted ovule, is known as anatropous ovule, and this type of ovule is found in members of renales. The third important type of ovule is compilotropous. Here you can see the image that is showing a compilotropous ovule. In this case, the ovule is more or less bean shaped, and in this case micropile also faces placenta. This type of ovule is found in members of Caryophyllaceae. The next type of ovule is amphitropous ovule. In this type of ovule, funicle is adnate to ovule for about half of its length. In this case, micropile faces laterally, and the placement of the ovule is slightly or somewhat right angle to the funicle. This type of ovule is known as amphitropous ovule. Finally, we have circinotropous ovule. You can observe, you can see in the image that in this case the ovule gets completely inverted. This curvature of the ovule or funicle continues till the ovule turns completely, And in this case, you can see here that micropile faces once again upwards. Such type of ovule is known as circinotropous, and it is found in Opuntia. Next, we have to discuss about the development of the ovule, that how ovule develops from mother primordium. Ovule primordium, ovule initials, appears as a protuberance on the surface of placenta. Then this protuberance grows into a conical structure which forms nucellus. From its base, one or two layers of tissues develop, which grow and form integuments. Outer integuments and inner integuments, but out of these, inner integuments grows first, then the outer integument. Then ultimately, with the passage of time and with the growth, these integuments enclose the whole portion of the nucellus except at the tip. That tip ultimately forms a micropile. All this development you can observe in this figure. This figure is showing ovule development in a very important plant, that is Arabidopsis. Here you can observe that how precursor molecules for the ovule, they grow and develop an ovule. We have talked about the ovule precursor molecules, that they grow in the form of a protuberance on the placental surface. You can observe here that protuberance, and that ultimately converts to a conical structure. Here you can see this NU represents the nucellus tissue. Then from the base of this nucellus tissue, there arises two more layers, which are represented by OI and double I here. OI represent the outer integument, and this double I is showing the inner integument development. These integuments grow and they completely encloses the central portion of the nucellus, except at the tip. This tip ultimately forms the region which is known as micropile. So this is a generalized picture for the development of the ovule. Parts of the ovule, basically nucellus and outer and inner integuments, along with the funicle and all other structures. Here we have another image that is showing the complete development of the ovule, along with the development of this central portion which is known as embryo sac. Here you can see that these are the integuments which are enclosing this nucellus portion, and this nucellus portion contains an inner region where megaspores are present. These megaspores are 4 in number, and out of these 4, 3 here degenerate, and that leaves only one megaspore. This single megaspore divides further and forms two spores, and in the next division two to form four, and in the next third division these four cells form 8 nuclei. These 8 nuclei, which are produced by the successive divisions of the original megaspore nucleus, they become rearranged in the structure in the form of megagametophyte. So this is the structure which is showing that how the ovule develops, and how the internal portions of the ovule, which is known as megagametophyte, develop from megaspores or megaspore mother cells. Okay, this was a complete picture that showing the development of the ovule along with all its parts. Next we have references. Thank you.